You are live.
Okay. Would you like me to introduce you now?
Yeah, sure.
Okay. All right. So I'm happy to introduce Maryam. Maryam is going to tell two scientific stories, and I want to briefly tell a third.
You are live.
Okay. You can hear me now, right? Would you like me to introduce you now?
Yeah, sure. I think someone has the video up.
So I'm happy to introduce Maryam.
I'm sorry.
Maryam is going to tell two scientific stories.
Okay. I am sorry. You can start.
All right. Are we good now?
Yeah.
Okay. So when Maryam first came to the lab, I asked her to investigate the redox control of NADH dehydrogenase 2. That was her first project. We had observed that the activity of this enzyme responded to the redox state of the cell, and surmised that there was likely to be a second redox cofactor in the enzyme that controlled the activity of its turnover. At that point, the literature had proposed that copper was an accessory metal in the enzyme. Maryam went on and proved that that was not the case. Unfortunately, contemporaneous crystal structures appeared at that time, which confirmed her result, but also scooped it. I then suggested that perhaps cysteine residues adjacent to the active site flavin were responsible for turning the enzyme on or off, depending upon whether a disulfide bond was formed. Bob Guinness provided mutants to Maryam that lacked those cysteine residues, and Maryam showed that I was wrong, that the cysteine-free enzyme still showed redox control. And at that point, we inferred that hysteresis was involved. That is, that when the enzyme had an oxidized flavin, it could transform into a conformer that was low in activity. Unfortunately, this is difficult to show, and it was certainly outside my skill set. So with some reluctance, I asked Maryam to abandon that project. And this was well into her time in the program. It was shortly before her prelim exam. So the project that then she picked up was one where she investigated how it is that mutants of E. coli that lack the known scavenging enzymes still could scavenge hydrogen peroxide at a low rate. I wasn't sure she'd be able to get the answer, but she did, and that's going to be her first story. When she finished that, she looked at a second thing, which is why there are many organisms that can't grow in the presence of virtually any oxygen at all. This is a fundamental principle in microbiology. It frames microbial ecology in lots of situations, including among human pathogens. And so she has an answer to that too. That's her second story. And those two stories actually comprise the bulk of what I talk about. Who that is. Okay. So she's had a big impact, not only on what her lab does, but on the field in general. And I also want to point out that she's made a significant impact on the community here on campus. She served in graduate college committees where she's given voice to international students in particular. She started the Graduate Student Association of Molecular and Cellular Biology, which now plays a significant role in our graduate program. And she did all this under fairly challenging personal circumstances, because we live in a difficult political moment, and that has precluded her visiting her family for quite a number of years back home. Yet she soldiered on in doing this. And so maybe it's sort of appropriate that she would be the first one to present a thesis defense entirely online. And so I'm gonna let her do that now. Okay, Maryam?
Yeah. Hi, everyone. Hi, Maryam. You probably know me, and I want to thank Dr. Imlay for his nice introduction. I will share my screen with you, which has my PowerPoint presentation, and I will walk over it. If you had any questions, please just comment them at the bottom of the video, and I don't know if I may be able to go over them and answer them at the end of the presentation. But if not, I would definitely go back and answer them after my thesis is over. So let's start this. Okay, so my work in Dr. Imlay's lab is basically on oxidative stress in anoxic habitats. That's the major theme that connects both of my projects together. The focus of the work in his lab is on oxidative stress in general, and we use E. coli as our model organism for most of our work, but at the same time, we use Bacteroides species, B. theta, to investigate some of the work that are done in obligate anaerobes. And I have one story in each of these bacteria, and I will just start with a very brief intro to oxidative stress. Why oxygen? What makes it special and unique, and what makes it be able to do what all the things that we know it can do? Oxygen has 2 unpaired electrons, but what is unique about them is that these are in 2 different orbitals. That makes oxygen a rare molecule, a rare type of radical, which is called a biradical. It has 2 results. First is that oxygen can accept 2 electrons. And the second is that it can accept them only one by one. And that is why we are just sitting in our chairs and we do not undergo spontaneous combustion as organic matters. When oxygen gets its first electron, it's converted to superoxide. When it gets the second electron, it's converted to hydrogen peroxide. When hydrogen peroxide gets the third electron, like the other electron, it is converted to hydroxy radical, which is here. And this is the most dangerous of all these 3, which are overall called reactive oxygen species, or ROS. Inside the cell, ROS is generated when oxygen encounters the flavins in non-respiratory flavoenzymes. These flavins are willing to give away their electrons, so it's easy for oxygen to take the electrons away and be converted into superoxide and hydrogen peroxide. With this very brief intro, I will go into my first story, which is the E. coli story. E. coli, as most of you probably know, is a facultative anaerobe. It's a gram-negative bacteria. It lives adjacent to the epithelial layer of our intestine. And it has been used as a model organism for years and years to investigate different matters, including oxidative stress. What we know so far about oxidative stress in E. coli is just summarized here, and I will go over it briefly. In its habitat, which is in the intestine, and during the time that E. coli is being transferred between hosts and it's out in the open, it experiences different types and different levels of oxidative stress. These oxidative stress can be the result of that can be endogenous, the result of reaction of oxygen with its own flavoenzymes. They can be the result of reaction of oxygen with reduced metals and thiols, or redox cycling compounds that are secreted by other bacteria or plants for defense. It also experiences hydrogen peroxide secreted by lactic acid bacteria and macrophages. So overall, they would E. coli would encounter superoxide and hydrogen peroxide that can go and in turn cause problems inside the cell. Perhaps the main one is that they increase the amount of unincorporated iron in a cell. It is important because that unincorporated iron and hydrogen peroxide can then react to form hydroxy radical, which I mentioned in the beginning of the talk is the most dangerous form of ROS, and it can readily damage DNA. These hydrogen peroxide and superoxide can also damage ionophore clusters and mononuclear iron enzymes. But the cell has different ways to deal with almost all of these problems to prevent them from happening or to repair the damaged moieties. And that would ultimately result in relief of oxidative stress. But perhaps the first step in all of these defenses is the presence of scavenging enzymes. Scavenging enzymes are enzymes that can degrade superoxide or hydrogen peroxide. E. coli has superoxide dismutase that can degrade superoxide, and it has 3 enzymes, alkyl hydroperoxidase AHP, and 2 catalases, CatG and CatE, that are capable of degrading hydrogen peroxide, which is here. They act in different conditions and under different circumstances. Now, my project started with an observation. We have a strain in the lab that is called HPX- strain. This strain lacks the 3 enzymes that I just mentioned, can degrade hydrogen peroxide, lacks CatG, CatE, and AHP. I wanted to, and we had observed that these cells can still degrade a little bit of hydrogen peroxide. So to further test that, I went ahead and measured the rate of peroxide scavenging in these cells and compared that to the wild type strain, which does include all these 3 genes. And in this assay, I added hydrogen peroxide to the cell culture, and I measured the amount of peroxide over time, and I measured the rate of peroxide degradation in the media. What we observed was that if I have the wild type cell that has these 3 enzymes, the rate of peroxide scavenging is high as expected. And in HPX- enzyme strain, that does not have these 3 scavengers, the rate of scavenging is pretty low. And this is if I grow the cells in Anoxic environment, which is basically in a water bath outside in the lab, when the cells are exposed to normal air that has about 20% oxygen. Now, if I grow the cells in anoxic conditions, we have an anaerobic chamber in the lab, and there's very low, under 50 ppm oxygen in it. And if I grow the cells in that condition, and I repeat the same experiment, the wild type strain is still, it seems like it's still expressing all these enzymes, and it can still degrade a lot of hydrogen peroxide. But our unexpected observation was that the HPX- strain showed a high degree of peroxide scavenging in these conditions. And I want you to note that this strain here, it does not have these 3 enzymes. So this means that there is an unknown scavenger that's expressed in anoxic conditions. This obviously brought up a lot of questions, including what is the source of this scavenging activity? How is it regulated? Is it there to protect the cell? And why does E. coli need a peroxide scavenger in anoxic conditions? Because I mentioned in the beginning that hydrogen peroxide is made from oxygen. So why do you need to degrade peroxide where there is no oxygen? To find the source of this scavenging activity, we dig the literature, and I found 8 different enzymes that have been reported to be able to degrade hydrogen peroxide outside the cell. So none of these enzymes had been tested in vivo or inside the cell. And I deleted them one by one in my HPX- cells, and I measured the rate of peroxide scavenging. The idea was that if any of these enzymes were responsible for this activity, if I deleted that, then the activity would go down. And that was what we observed. So these are the list of my different enzymes. And here is my parent strain, which is the HPX minus strain. And you can see that when I deleted CCP, the scavenging activity went down. So it suggested that CCP is the anoxic hydrogen peroxide scavenger. CCP was previously known as YHJA in the literature, and we suggested in our paper to rename it to CCP. It is a triheme cytochrome C peroxidase. It has 3 subunits. Each subunit has 1 heme. It is a periplasmic enzyme, and it gets electrons from somewhere and puts them on hydrogen peroxide to reduce that to water. This is basically all we knew, almost all we knew about this enzyme at the point we started this work. So we started characterizing it. Our first question was, where is the electron coming from? Because this enzyme was placed in the periplasm. Our first guess was obviously the electron transport chain. And we know that electrons are transferred in the electron transport chain using quinones, either ubiquinone or menaquinone. So I deleted either of these quinones, and I showed that the rate of peroxide scavenging in menaquinone minus strain is lower. That suggested that, yes, this enzyme is actually getting its electrons from the electron transport chain, and the electrons are being transferred by menaquinone. And we know that menaquinone is the main quinone in anoxic conditions, so that made sense. The second question that we wanted to answer was, how is the electron transferred from this quinone pool to our enzyme? I did a lot of experiments. I deleted a lot of different complexes that are shown to be able to mediate this transfer in the other conditions. But ultimately, we came to the conclusion that this transfer is probably a direct transfer, meaning that CCP directly gets the electron from the quinone pool. And that was what we published in the paper. And about a year later, another paper came out, and they showed that CCP or YJA is indeed a membrane-bound protein that is embedded in the inner membrane, and it directly gets electrons from the quinones. And ultimately, we wanted to make sure that CCP is there to degrade hydrogen peroxide, and hydrogen peroxide is its real substrate. And we show that by measuring the affinity of the enzyme for the substrate, or the Km. And I measured the Km for this enzyme, and it turned out to be about 5 micromolar, which is low. So, and overall, our data showed that CCP receives its electrons from the electron transport chain, and it has a high affinity for hydrogen peroxide. But how is it regulated? At the time I started this work, there was only one other paper published on CCP in E. coli. And in that paper, it was shown that the promoter of this enzyme has binding sites for 2 transcription factors called OxyR and FNR. What are they? FNR responds to oxygen. In the presence of oxygen, it is in monomer form and it's oxidized. When oxygen is gone, it forms a dimer and it can turn on the multiple genes that are all in the FNR regulon. OxyR responds to presence and absence of hydrogen peroxide. In the absence of hydrogen peroxide, it is in the reduced form. In presence of hydrogen peroxide, it gets oxidized and it can turn on genes in the OxyR regulon. Okay. So I wanted to confirm this and repeat these experiments. And what I did was that I made a reporter, and by adding the promoter of CCP to the reporter gene LAC-Z to be able to measure the expression of this enzyme easily. And you can see here that if I grow the cells in oxic environments, just in normal air, it doesn't matter if I add hydrogen peroxide or not, I do not see much expression from this enzyme. If I grow the cells in anoxic environments without hydrogen peroxide, I just see a little bit of expression. And the highest level of expression is seen in anoxic conditions in presence of hydrogen peroxide. To confirm that this role is done by FNR and OXCR, I used FNR and OXCR mutants and used the exact same condition that otherwise in wild type gives me full activity, but these mutants do not, full expression, but these mutants cannot express this enzyme, even when either of them are gone. So this showed that CCP is regulated by both FNR and OXCR. It needs both of them to be activated, and it's transcribed when hydrogen peroxide is present and oxygen is absent. But where would bacteria encounter hydrogen peroxide but not oxygen? We think that this special phenomenon happens on the what we call the oxic-anoxic interface. For E. coli, for example, that lives in the gut, we know that the cells are, we know that oxygen penetrates through the epithelial layer into the lumen, okay? And it produces a gradient that here is shown with orange color because there are all these bacteria that quickly consume oxygen. But towards the center of the lumen, we have sulfur-reducing bacteria that produce H2S. Now we have H2S coming from one side and oxygen from another side. And oxygen can easily steal the electrons from H2S and form hydrogen peroxide. And this oxic-anoxic interface is where we think E. coli lives and where it experiences hydrogen peroxide, but very low levels of oxygen. Now, I just showed you that E. coli probably lives in an environment that has hydrogen peroxide, and it is expressing a special enzyme in that condition. So the first thing that comes to the mind is that it is there to protect the cell from the peroxide that's coming from outside. We did a lot of different experiments, but I'll just show you one modeling that we did. And in that, what we did was we used the transfer rates between the membranes and different like scavenging rates for hydrogen peroxide. And we showed that when hydrogen peroxide enters the periplasm from the environment, most of it goes back out through the porins that are in the outer membrane. About 2.7% of it can pass through the inner membrane and get into the cytoplasm. This was modeled without having CCP in the picture. Now, if I add CCP to the picture, you can see that it degrades, and assuming that CCP is fully expressed and it's at its full activity, it scavenges a portion of this hydrogen peroxide that is in the periplasm, but the amount of peroxide that gets into the cell is now 2.6%, which is only 0.1% different from when we did not have CCP. So CCP does not protect the cytoplasm from exogenous hydrogen peroxide. Then we had an alternative hypothesis. Does CCP enable E. coli to use hydrogen peroxide for respiration? What I will just give you a very brief reminder about anaerobic respiration. So we know anaerobic respiration happens when we have electrons entering the electron transport chain. The source of the electron can be NADH, formate, hydrogen, different molecules. The electrons are then transferred in the electron transport chain. There can be different proteins in between, but the transfer normally happens through quinones. And ultimately, the electron is transferred to an electron acceptor at the end. Now, if the electron acceptor is oxygen, we will have aerobic respiration or aerobic respiration. If the electron acceptor is not oxygen, in this case, for example, if you have nitrate or fumarate, then this respiration is called anaerobic respiration. we had a clue that told us that we are probably in the wrong path, in the right path. Again, when I used the BlackZ reporter to measure the expression of my gene, I showed that adding fumarate does not affect the expression of this gene, but if I add nitrate or oxygen to my culture, the expression of CCP is inhibited. Okay? So both oxygen and nitrate inhibit the expression of this enzyme. But what does it mean? To put it into context, I will show you the preference of E. coli for respiratory substrates. We know that E. coli likes oxygen more than it likes nitrate and more than it likes fumarate. Now, nitrate reductase, which is needed for using nitrogen, for using nitrate as the electron acceptor, is expressed only when oxygen, which is the better substrate, is absent and when nitrate is present. Fumarate reductase is expressed only when oxygen and nitrate are absent, which are the better substrates, and fumarate is present. And cytochrome C peroxidase, CCP, the enzyme that I was working on, is expressed only when oxygen and nitrate are absent and hydrogen peroxide is present. So it really beautifully fit into this respiratory substrate scheme that we had from before. So what I was trying to, so we wanted just to see if our hypothesis is true, if hydrogen peroxide can be used as a respiratory substrate. To test that, I used glycerol. Glycerol is a non-fermentable carbon source, meaning that it cannot be fermented and the cells need to run an active electron transport chain to be able to use glycerol as their carbon source and grow on it. In this form, glycerol would be converted to glycerol 3-phosphate. The electrons would be transferred to the electron transport chain. And now what we try to check is that whether this reaction is happening, whether the electrons can be put on hydrogen peroxide, and that would result in this path of electrons from glycerol to hydrogen peroxide going, and that would enable the cells to grow. So to test that, I added hydrogen peroxide to my media. So here I show that if I, so this experiment is done in the absence of oxygen. So there is no electron acceptor. If I do not add any electron acceptor to my media, nothing is there and the cells do not really grow. If I add nitrate, which is a very good electron acceptor, to the media, they grow fine. If I add fumarate, they also grow. And if I keep 5 micromolar hydrogen peroxide in the media, the cells can also grow on it. So yes, E. coli grows on peroxide as the sole electron acceptor. To make sure that this is because of CCP, so I made a mutation. So here is the wild type strain. As I showed before, it grows in presence of peroxide and not without peroxide. But if I used the CCP mutant that lacks the CCP gene, then it didn't matter if I added hydrogen peroxide or not, the cells could not grow. So this suggests that the growth on hydrogen peroxide is mediated through CCP. We also wanted to make sure that the phenomenon that we are seeing is not because we are using a very common E. coli lab strain. So we tested that on E. coli B, which is not as used as much in labs. And I showed that, yes, if I added hydrogen peroxide, E. coli B also grows. And I also tested that on Salmonella. And Salmonella also grows on hydrogen peroxide. So this seems like a broader phenomenon that's happening, and it's not only restricted to E. coli. Now, our final model at this point is that we have hydrogen peroxide in the environment. As I mentioned, E. coli lives in the oxic-anoxic interface, so we expect it to experience oxidative stress and hydrogen peroxide. The peroxide gets into the periplasm almost freely. A portion of that passes the inner membrane, and some of that gets degraded by the scavenging enzymes, catalases, and peroxidases. Now, at this point, if we have about 0.2 micromolar hydrogen peroxide, that is enough to turn on OxyR. And if this process happens in the absence of oxygen, then we have FNR turned on as well. And together, they will bind to the promoter of CCP. They would express the protein, and it would be transferred to the periplasm. And at that point, if we have the electron transport chain, the electrons can be transferred from different sources here. For example, I've shown NADH to quinones. And from there, ultimately, CCP would be able to take the electrons from the quinones, put them on hydrogen peroxide, and convert that to water. And through this, it can allow this metabolism, this part of metabolism to keep going. Now, what I just suggested was adopting the toxic chemical for metabolism. What I'm saying is that hydrogen peroxide is toxic, gets inside the cell, and it causes all of those damages that I talked about in the beginning of the talk. And I suggest that it can be incorporated in the metabolism, and it can be used to the benefit of E. coli. It may sound a little bit weird, but it's not totally that weird, because it has happened before, about 2.4 billion years ago, when cells started to adapt to presence of oxygen. Oxygen is toxic when O2 itself is toxic when it gets inside the cell. But the cells somehow managed to be able to incorporate it in their metabolism and start all this oxic life that we are seeing all around us, including us humans. So it might not be a very crazy idea at the end. So this was the end of the first part of my talk. If you have any questions about that, please just comment them below, and I'll see if I can get back to it. Now, I will start my second story, which is the B-theta story. Excuse me. I'll start by the atmospheric oxygen. Atmospheric oxygen was not 20% throughout the history of the Earth. Okay. What we know at this point is that after the Earth was formed somehow, and the first life started again somehow in the oceans, life started in a world that was devoid of oxygen. And the cells that were living at that point did not have to worry about dealing with oxygen or reactive oxygen species. But with the evolution of oxygenic photosynthesizing microbes, the amount of oxygen started to raise in the oceans and then in the atmosphere, as it can be seen by the red line here. The cells kind of took two different approaches at this point. Some learned how to survive in presence of oxygen, how to grow in presence of oxygen, and how to ultimately use oxygen to their benefit to be able to take more energy from their food. Okay. Other group of bacteria or other group of cells basically remained in the anoxic environments that were still available in the oxic world. Okay. And these are what we call the obligate anaerobes. Okay. The phenomenon of obligate anaerobiosis has been a very interesting phenomenon, and many people have been working on it over the years. Because as we know, if you are using the same carbon source, okay, for example, if you're using glucose, if you are adapted to using oxygen in your metabolism. If you do it aerobically, you can make about 20 times more energy than if you do it in the absence of oxygen. So the big question was, why are obligate anaerobes obligate anaerobes? What is stopping them from leaving their anaerobic environments that does not have oxygen and explore the oxic world to be able to take more energy from the food? Okay. And a lot of work had been done on this. A very important paper came out in 1971 by John McCord and Irvin Friedovich. What they did was that they selected a couple of obligate anaerobic bacteria, and they they checked if these bacteria have catalases or superoxide dismutases. If you remember, I mentioned catalases degrade hydrogen peroxide and superoxide dismutases degrade superoxide. What they came to was that these obligate anaerobes do not have catalases or superoxide dismutases. That resulted in them suggesting an enzyme-based theory of obligate anaerobiosis, which stated that obligate anaerobes are obligate anaerobes because they do not have these scavenging enzymes, and therefore they cannot deal with the adverse effects of superoxide and hydrogen peroxide in the oxic world. So they just remain in their anaerobic world to prevent that. But more work was done, and it was shown that these people were probably kind of unlucky in their choice of obligate anaerobes. It was shown that these obligate anaerobes that were checked, they had peroxidases to degrade hydrogen peroxide, not catalases, and they had superoxide reductases and not superoxide dismutases. And because the assays that they were doing were specific for those special enzymes, they could not detect the other activity at that time. And papers came out after this, and they showed that, and what we know at this point is that virtually all organisms contain enzymes to degrade hydrogen peroxide and superoxide and to defend themselves against these. Then another question was justifiably brought up, and that was, why don't they just ramp up their ROS scavengers at this point? So if they do have these enzymes, it means that they can deal with hydrogen peroxide and superoxide. Then they can just increase the amount of their catalases and peroxidases, for example, and then they can enter the oxic world and explore it. Okay, why don't they do it? These two main questions are what we sought to answer in the second project that I did. And in this project, I used an obligate anaerobe. It's called Bacteroides theta iota micron. The Bacteroides genus in general is the second most abundant genus of bacteria after firmicutes in the gut. They are particularly important because they are capable of degrading complex sugars and turning them into simple sugars that can therefore then be used by other bacteria in the gut. Basically, Bacteroides trash is other bacteria's treasure at this point. And what we know is that B-theta is an obligate anaerobe. Okay. And what we know about obligate anaerobes is that they stop growing in presence of oxygen. So I showed this here. If I grow these cells in the absence of oxygen in our anaerobic chamber, they grow fine. They don't have a problem growing. If I take the flask that contains these cells out and put it in like 20% air that contains 20% oxygen, and at 37 degrees, these cells stop growing in presence of oxygen. What is surprising is that if I take the cells after I aerated them for almost more than 2 hours, if I take the cells back in the chamber, I change the media to anoxic media, and I let them, and I put them at 37 degrees in the anaerobic chamber, these cells almost rapidly start growing. So this tells us that during this phase where the cells are not really growing as much, they are alive. And it was shown before in our lab through CFU measurements that the cells do not die in this process. But then the question is, why do they stop growing then? Okay. And it seems like that something is blocked. They are just sitting there. The metabolism is blocked, probably, but we don't know what the problem is. So it was to answer that, we would need to take a look at the central metabolism of B theta, and I will walk you through that. It starts with glycolysis, where NAD is converted to NADH. We will end up with phosphoryl pyruvate, where the branching of metabolism happens in B theta. In one branch, we have what is basically the reverse half cycle of TCA, where phosphoryl pyruvate is converted to oxaloacetate, malate, fumarate, and succinate. And throughout this process, what is important is that we will regenerate NAD. Okay, and I showed you that NAD is used up in glycolysis in the first step. Okay, so that would so the left branch here is needed to basically keep a cycle of NAD and NADH going. And here I need to put 2 NAD and 2 NADHs. So there is a balance because we have we need two NADs, and one is produced here and one is produced here. Okay. On the other branch, we have phosphoryl pyruvate that's converted to pyruvate, and used 2 different enzymes convert pyruvate to acetyl-CoA here. Okay, they can be B theta has PFL, or pyruvate formate lyase, that does this conversion. So pyruvate has 3 carbons, acetyl-CoA has 2 carbons, the acetyl part has 2 carbons, and the extra carbon is released in form of formate. Okay. The other way to come to the same result is using PFOR, pyruvate formate pyruvate ferredoxin oxidoreductase. Okay. The extra carbon is released here as form of CO2, not formate like there. And in the process, 1 molecule of ferredoxin is reduced. Then that can then later be oxidized back through different enzymes, for example, hydrogenases. What is important here is that acetyl-CoA is necessary for formation of fatty acids. And fatty acids are necessary for making membranes, and membranes are necessary for cell growth. So if the cells cannot produce acetyl-CoA, they cannot grow. Therefore, the main products of the left branch, the main product is NAD, and the main product of the right branch is acetyl-CoA. It was shown in our lab previously that 3 enzymes in this central metabolism actually are damaged, and they are inactivated in B theta cells that are aerated. If you remember the graph that I showed you, when the cells grow and then they do not grow, during that phase where they are not growing, it was shown that these 3 enzymes are damaged in them. And these are PFL and PFOR and fumarase. But what are these enzymes? I will just briefly introduce them to you. And I know going back and forth between 3 enzymes can be a little bit confusing. So up here I have small scheme. The enzyme that I'm talking about would be yellow. So if anytime during the talk you felt lost, you can just refer back to that. PFL, as I mentioned, is pyruvate formate lyase. It converts pyruvate to acetyl-CoA. It releases a molecule of formate. What is unique about this enzyme is that it has a glycyl radical in its active site. The glycyl radical is formed by PFL activating enzyme from a regular glycine residue in the cell, in the active site of the enzyme. Now, If you remember, I told you that oxygen is a radical. It's a biradical. Okay? Radicals and radicals, when you put them together, bad things happen. And here, what happens is that the radical oxygen would come react with this radical, gaseous radical that is here. That would ultimately result in the polypeptide chain to break in half at the point of the where this gaseous radical is present. And this can be seen beautifully here. On the left, you have pyruvate formate layers that is not exposed to oxygen. And on the right, if you expose the purified protein to oxygen, you can see that it is cut in half, basically. It was shown that PFL is extremely sensitive to oxygen, as I explained to you why, and it loses almost all of its activity in 20 minutes. Okay. And the oxidative stress that is caused here is by O2 itself, not by superoxide, not by hydrogen peroxide, O2. Okay. The second enzyme is fumarase. Fumarase, as I mentioned, is it is involved in conversion of fumarate to malate. And the two postdocs in our lab, Zheng Liu and Ramki Sethu, they showed that fumarase is inactivated in B-theta in about 1 hour. When you aerate the cells and measure the activity of fumarase, almost all activity is gone within 1 hour. And they also showed that this damage is the result of superoxide. So here, superoxide is the moiety that is the ROS that is damaging here. But the weird observation that they had was that while in B-theta, the fumarase loses activity in 1 hour, in E. coli that also contains fumarase, fumarase stays active for hours, like even after 3 hours of aeration, it still stays active. Then they asked the question, why do enzymes die in B-theta but remain active in E. coli? To test that, there are two possibilities here. First is maybe there is something that is intrinsically different between the E. coli enzyme and the B-theta enzyme. So maybe the B-theta enzyme itself is more sensitive to oxygen or to superoxide. And the second option was maybe the environment at which these enzymes are, which are the B-theta cell and the E. coli cell, are different somehow when the cells are aerated. To test that, what they did was that they expressed the E. coli gene, the E. coli fumarase in B-theta, and they expressed B-theta fumarase in E. coli. If the enzymes, B-theta fumarase or E. coli fumarase, are in E. coli, they both stay active for 3 hours. Okay. And if they are expressed in B-theta, both of them lose activity in within an hour. So this suggests that something is different between the environment of the cell in B-theta versus in E. coli. And we know that the cells produce reactive oxygen species when they are aerated. And to see if there is a difference between the amount of ROS that's produced in these cells, what they did was that they measured the rate of ROS production in E. coli, which you can see here, and in B-theta. And if you look at the slope of these two graphs, they came to the conclusion that aerated B-theta generates 15 times more ROS. Here it was hydrogen peroxide that was measured than E. coli, which is a very big difference. It means that there is an enzyme, or there are enzymes that are so willing to give away their electrons that when oxygen comes around, it can easily take the electrons away and produce hydrogen peroxide and superoxide. to find the source of this, what I did was I went back to the central metabolism scheme that we had, and I deleted different parts of this scheme and different genes and different proteins that are involved here. And making these mutants were not the easiest thing. Here is me with a couple of plates that I used to make one of these mutants. But the idea then was that I would delete these genes, and then I would aerate these cells. Now, if, for example, PFL, which is here, if PFL is responsible for the production of ROS in aerated B theta, if I delete it and expose the mutant to air, then the amount of ROS that's produced in that mutant would be lower. So I did all the experiments and tried to see in which mutant produced less ROS than the parent strain. The answer was none of them. Okay. At this point, I think it is okay, just to put this into perspective. We do not know the source of ROS production, like the full source in any organism. We do not know it in any bacteria, in any archaea, in any eukaryotes. The only clue that we have is we know about the source of about 30 to 40% of ROS production in E. coli, and that is all. So this is not an easy problem to tackle. It's been taking years and years, so we would pursue that. But obviously, the day it happened, that was not the best day of my life, but I think I had to move on. Now, I talked about PFL. I talked about fumarase. The third enzyme is PFR, or pyruvate ferredoxin oxidoreductase. PFL, as I told you, converts pyruvate to acetyl-CoA. It produces CO2. It produces reduced ferredoxin that can then be, in turn, be oxidized using enzymes such as hydrogenases or RNF. Now here is the resolved structure of this enzyme from the sulfovibrio. And you can see beautifully that it has 2 subunits shown in blue and red here. Each subunit has 3 iron sulfur clusters. They are yellow and orange, and 1 TPP, which is red and gray and blue. And the pyruvate, which is the substrate, binds somewhere around here. Okay. Then the electrons are transferred from pyruvate to TPP, and from there they hop to the first iron sulfur cluster, to the second iron sulfur cluster, to the third iron sulfur cluster, and from there they hop to the surface where ferredoxin, oxidized ferredoxin, is ready to take the electron away and become reduced. So this is the electron transfer path for PFOR. Now, what activates, what I have shown so far is that PFL is inactivated by oxygen. Fumarase is inactivated by superoxide. But what is not known is the ROS that inactivates PFOR. So this is what I try to go to and answer. To do that, I measured the rate of PFOR inactivation in a couple of different strains. One of them is the wild type strain, obviously, which is the parent. One is HPX minus. This strain lacks the enzymes that degrade hydrogen peroxide. The third strain was SOD minus, which lacks the enzymes that the enzyme that degrades superoxide, which is superoxide dismutase. I also used a strain that I call PSOD. It has superoxide dismutase on a plasmid, and it overproduces superoxide. So the idea is that the wild type strain would have a normal amount of ROS in it. The HPX minus strain, because it does not have hydrogen peroxide scavenging enzymes, it has way more hydrogen peroxide in it when they are aerated. The SOD- strain, because it does not have superoxide dismutase, it has way more superoxide in it when the cells are aerated. And the PSOD strain, because it has a lot of superoxide dismutase, has way less superoxide in it than the wild type. So they all have different amounts of superoxide and hydrogen peroxide in them. But what was interesting was that the rate at which PFOR lost its activity was the same in all of them. That suggested that hydrogen peroxide and superoxide are not the species that are inactivating PFOR. To look into this a little bit more, I incubated whole cells of B-theta and incubated lysate of B-theta, and I incubated purified PFOR protein with different concentrations of hydrogen peroxide, up to 1 millimolar. And you can see here that these red boxes, they are, I try to capture somewhere around 10 micromolar hydrogen peroxide. That is what we believe is B-theta is going to experience in its environment. So that is like the physiological concentration of peroxide for B-theta. And you can see that the activity of PFOR is almost the same in all of these conditions if I only use 10 micromolar hydrogen peroxide. Therefore, it further confirms that physiological doses of peroxide cannot inactivate PFOR. Now, we showed that PFOR inactivation is not dependent on hydrogen peroxide, but on the concentration of hydrogen peroxide, but is it dependent on concentration of PFOR? To test that, I incubated whole cells in buffers that contain different amounts, different levels of air saturation. So 100% air saturation means that there is air with 20% oxygen in it. And I showed that, yes, indeed, PFOR inactivation correlates with oxygen concentration. The last piece of evidence that we had at this point was from the structure of this enzyme. This is what I showed you before, and this is the space fill model of the same part of the enzyme. And I want you to notice that these orange and yellow iron sulfur clusters that are here, they cannot be seen here. It means that they are not exposed to the solvent. They are not exposed to the outer environment of this enzyme. Why is it important? We know that hydrogen peroxide and superoxide, they need to directly bind to whatever molecule they are oxidizing. So in this case, if we consider the iron sulfur cluster, for example, in the case where the cluster is inside, deep buried in the protein, and the hydrogen peroxide is outside, then there's a distance between them, and therefore electrons cannot be transferred to it because there is no direct interaction. The same is true for superoxide. Again, if superoxide is out here, the electron transfer does not happen. But what is different about oxygen is that oxygen is capable of what we call outer sphere electron transfer. It means that electrons can basically hop to oxygen without it being directly bound to its target. Therefore, if the cluster is inside here and we have oxygen here, the electrons can basically hop from the cluster to oxygen. And it does happen indeed. So this shows that from structural evidences, it is also evident that superoxide and hydrogen peroxide cannot be inactivating PFOR. So overall, these put together show that PFOR inactivation is done by O2 itself. But so going back to the first model, I showed you that we know that the inactivation is done by PFL. The inactivation of PFL is done by oxygen. Inactivation of fumarate is done by superoxide. And I showed that inactivation of PFOR is done by molecular oxygen itself as well. Now, what does it mean? to the cell, which of these activities are essential. To test that, I made mutants. These are different from the other mutants that I made. Okay, they're in the different background, so all over again. But I deleted these genes again one by one, and in some cases I made double mutants, and I measured the growth of these mutants. Now, as you can see, if I delete fumarase, which is here, okay, in the left branch, the cells cannot grow. If I delete PFL alone, it is one of these lines, okay, I assure you, they grow. If I delete PFL, the cells can convert pyruvate to acetyl-CoA through PFOR. Okay. If I delete PFOR, the cells again can grow because they can convert pyruvate to acetyl-CoA through PFL this time. But if I delete both of these, therefore, if I block the whole right branch, then the cells cannot grow at all. Okay, so B theta needs both of these branches to grow. To dig deeper into this, I did an NMR analysis of the fermentation products of these mutants. So I grew these cells, and then I did NMR to find out what are the molecules that each of these mutants secrete out to the media. Here you can see them, and I want to walk you through this. If you look at the acetate level, so the acetate secretion is a result of the formation of acetyl-CoA, okay, because acetate can be formed from acetyl-CoA. And you can see that in all of these peaks, the only two strains that do not produce much acetate, therefore do not produce much acetyl-CoA, have PFR mutation. So this has PFR mutation and this has PFR mutation in it. And both of these strains, they show a new peak, which is the pyruvate. It suggests that if you delete PFOR, yeah, the cells can use PFL, but it seems like PFL is not as good in degrading, in converting pyruvate. Therefore, pyruvate starts accumulating in the cell and it gets secreted into the environment. These mutants also secrete a lot of formate. That makes sense because if we delete PFOR, it means that the flux through PFL goes higher, therefore more formate is produced. But if you look at all the other mutants, so I deleted a lot of different things here, but all of the other ones look almost exactly the same as the wild type strain, which is the parent, and it's the graph up here. So it seems like the metabolic pathway stays stable in all mutants except for PFOR. Okay. Now, we have a big picture view of what is happening here and what is important in this scheme. And this was all when I discussed about the time when B theta is not growing. Okay. But what happens when it resumes growing? Because if these enzymes are inactivated, then somehow they should be reactivated, we think, for the cells to resume growth when we put them back. So we looked at fumarase reactivation. What we know about fumarase is that fumarase has an iron sulfur cluster, and this iron sulfur cluster is exposed to the environment. Superoxide can come and steal and basically oxidize this cluster. That would result in the iron atom that was here to just fall off. Now, we want to see if the cells are capable of reactivating. Here, I show you, I took some B theta cells and I aerated them for 30 or 60 minutes. Now, if I keep them in an anoxic environment, nothing happens. But in the aerated cells, you can see that the activity of fumarase goes down almost all the way within 60 minutes. Now, if I take these cells and put them back in the anaerobic chamber, and if I change the media to anaerobic media, and I wait for 1 hour, for 2 hours, and for 6 hours, you can see that the activity of fumarase gradually resumes as it goes up, which means that the cell is able to put that iron back in here. Okay, so this whole enzyme can be active again. Another way for us to test it is to check whether our in vitro reactivation works. In vitro reactivation is when I have the lysate that contains the protein in it, and I add iron, because we know an iron is lost, with a reductant. Here I added DTT because we need to reduce the cluster to be able to put back the iron. And as you can see here, if I do the in vitro reactivation with iron and DTT for half an hour or an hour and a half, in these cases, I also get full reactivation of this enzyme. So fumarase can be reactivated both in vivo and in vitro. Now, can PFR be reactivated? PFR, as I showed you, the clusters in it are not exposed to solvent. Okay. So when they get oxidized, the iron probably falls off somehow, but this whole area is not accessible to the repair systems. When I did the previous experiment, I used the exact same lysates, and I measured the activity of PFOR in them. So again, we have the cells that were kept anaerobic for 60 minutes, they're fine. The cells that are aerated, you can see that, again, the activity goes down within 60 minutes. The activity of PFR goes down. Now, if I put the cells back in the chamber and I wait for 1 hour, 2 hours, or 6 hours, the activity does not come up. If I add iron and DTT to rebuild the cluster basically in vitro, the activity does not come out either. So this suggests that PFR cannot be reactivated in vivo or in vitro. Now, putting all of these things together, what I showed you, and summarizing all this, what I showed you is that we know that these three enzymes are damaged. PFL is damaged by oxygen, but my mutations showed, and growth curves showed that it is probably not the most important enzyme of these two. So its inactivation does not probably have much effect on the growth of the cell in our condition. Because when I deleted that, the cells were okay in their growth. Fumarase is inactivated, and it's necessary because when fumarase is inactivated, then the left branch is inactivated, and I showed that the cells need the left branch to grow. But what is good about fumarase is that it can be reactivated. So when we put the cells back in, they should be fine. Okay, so again, just as a reminder, this is the reactivation scheme of fumarase. It seems like the biggest culprit in the oxidative stress in B-theta is PFOR. PFOR, first, it is damaged by O2 itself. And second, it cannot be reactivated. Okay. So if you lose all the PFOR activity, it would be very hard for the cells to come back out. But if you remember, I showed you that activity of PFOR depends on the concentration of oxygen. And we believe that in normal conditions where B-theta experiences oxidative stress, and when it is exposed to O2 itself, it would probably be a lower concentrations, not a full 20% oxygen that is in the air. And at lower concentrations, it manages to maintain some activity of PFOR. Therefore, it would be able to keep the flux through the right branch, and the fumarase would keep getting inactivated, but it keeps getting reactivated. So we will have some degree of fumarase activity as well. So we will have the left branch running as well. Therefore, it seems like they would be able to survive these conditions. Now, going back to the first two questions that we had in the beginning, What are obligate anaerobes? Obligate anaerobes. Putting facts together, we know that obligate anaerobes are successful at dominating anoxic niches. In our gut, for example, which is almost in a fully anoxic environment, obligate anaerobes outnumber facultative anaerobes by 4 degrees of magnitude. Oxidative stress does happen in these conditions, and these cells have enzymes and have equipments to deal with these oxidative, with the situations of oxidative stress. But still, they cease growth in presence of oxygen. And we think the reason for that is that obligate anaerobes are oxygen sensitive because the best anaerobic metabolism, it is intrinsically vulnerable to oxygen. Right? And in the cases of what we studied, it could be because of low potential biochemistry, as we saw in PF4, where the electrons could be easily transferred from PF4 to oxygen, or using radicals, as we saw in PFL, because the radical in PFL makes it very vulnerable. But both of these are the enzymes that are needed in obligate anaerobes for their activity. And the second question was, why don't they just ramp up their RS scavengers? Now, if you remember, I showed you that the right branch of metabolism is fully, can be inactivated by O2 itself. It means that even if the cells have no hydrogen peroxide and no superoxide in them, the growth would still stop because O2 itself would go and damage PFL and PF4. Right? And therefore, the role of molecular oxygen in this phenomenon is probably more important than what we thought. That is the end of my second project. Again, if you had any questions, please just comment them at the bottom of the video. I'll get back to them. For this, I would like to thank the people who helped me all these years. Dr. Imlay, obviously, was way more than just a mentor. I learned science from him, and I learned how to be a scientist from him. I want to thank Imlay Lab. They are cool people. They are amazing. And I look forward to working with them all the time. I want to thank my committee, Dr. Cronin, Dr. Ganis, Dr. Imlay, and Dr. Metcalf. And they were always there writing recommendation letters for me and giving me strains left and right. So, and they were very supportive during all these years. And I want to thank my lovely family and friends. با مامان خوبم، خانواده گلم، دوستای قشنگم. مرسی که این همه مدت با من بودید، مرسی که حمایتم کردین و بدونید که خیلی دوستتون دارم، خیلی دارم براتون تنگ شده. قربونتون برم. With this, I will just end the live stream, and I'll go back to meeting with my committee, and I will comment the result. Yeah, so bye for now.